Hello, everyone. I'm so sorry that our session today will have to proceed asynchronously again. Um, there's been a lot of rescheduling, and therefore, a lot of my commitments have been compressed to the next following weeks. But hopefully, this is our last asynchronous session for lab for the entire term. So we are finally on our last module for genomics and proteomics lab. And this is the last module before your expert talk on our final meeting this term. And it's on gas chromatography and mass spectrometry, or GCMS. So GCMS is an analytical technique that combines two powerful methods, gas chromatography and mass spectrometry, to separate, identify, and quantify compounds in a sample, especially those that are volatile or can be made volatile. So here's how each component works. In the gas chromatography phase, or the stationary phase, a sample is first vaporized and then injected into a long, narrow tube known as a GC column, and an inert gas. So this is a carrier gas. It's usually helium or nitrogen. This inert carrier gas moves the vaporized sample through the column, which is coated with the stationary phase material. Each compound in the sample interacts differently with the stationary phase, which causes them to separate as they move along the column at different speeds. The time it takes for a compound to travel through the column, known as its retention time, helps distinguish it from other compounds. Now, in the second phase, the mass spectrometry phase, as each separated compound exits the GC column, it can now enter the mass spectrometer, where it is bombarded by high-energy electrons in the ionization chamber. So this bombardment breaks the compound into charged fragments. So these fragments are then accelerated into a mass analyzer, which sorts them based on their mass-to-charge ratios. So napag-usapan na natin yan sa lecture. The detector records the abundance of each fragment, producing a mass spectrum, which is a unique fingerprint of each compound that helps identify its molecular structure. So GCMS has quickly become a very important technique in proteomics because of its ability to accurately identify and quantify complex mixtures of compounds, even at low concentrations. So it's widely used in environmental analysis, in forensics, in food and beverage safety, in pharmaceuticals, and more. For example, based nagamit na ng lab na nagamit na siya ng lab namin to identify pollutants in the creek beside Brahms. So because of that, we were able to have discussions with nearby farmlands on their tendency to overload their soil with fertilizers that can pollute nearby bodies of water. But apart from that, GCMS is also commonly used to detect drug residues in blood and also verify pesticide levels in food. So for our exercise, these are our learning objectives. We need you to be able to understand the principles of GCMS so that you're at least familiar with how it works and how it's able to analyze chemical mixtures. We need you to be able to interpret GCMS chromatograms and mass spectra. So in this exercise, we'll give each group their own unique data set that you will need to analyze, and then apply the data that you got by creating a written report that is set in a real-world scenario, and then synthesize and communicate your findings. Okay? So let's first talk about the details on how GCMS is done. So for sample preparation, think this is the hands of Sir Jovi, one of my RAs. The sample is processed to extract the target compounds and remove interfering substances. So depending on the nature of the sample, we have various extraction methods such as liquid-liquid extraction or solid phase extraction, any of that could be employed. So proper sample preparation ensures that the compounds of interest are concentrated and is in a form that's suitable for analysis. So it minimizes yung tinatawag na matrix effects, which can affect the accuracy and sensitivity of subsequent analysis. So well-prepared samples basically lead to more reliable results. From there, we move on to the injection of the sample into the GC column. So this is when your sample is introduced into the GC system where it is vaporized. The injector port is maintained at a high temperature to ensure that the sample becomes a gas. The inert carrier gas then transports the vaporized sample through the system. So this step is crucial for transitioning the sample from a liquid or solid state to a gaseous state, allowing it to enter the GC column. The efficiency of the injection process impacts the amount of sample that reaches the column, which directly affects sensitivity and detection. From there, we move forward to the third step, which is the separation of compounds in the GC column. So the GC column is packed with a stationary phase that interacts differently with various compounds based on their chemical properties. So pwede kang mamili. Would it be based on their polarity or their boiling point? So as the carrier gas moves through the column, compounds are separated based on their affinities for the stationary phase. So this separation allows for the isolation of individual components from a complex mixture. So each compound's unique retention time, which is determined by its interactions with the stationary phase, helps in identifying and quantifying them in the sample. And then for step 4, that's the detection of compounds as they exit the GC column. So the GC system is equipped with a detector. So it can be a flame ionization detector or FID, or a thermal conductivity detector or a TCD. So this one measures the concentration of compounds as they elute from the column. The detector produces a signal proportional to the amount of each compound present. So this step generates a chromatogram, which is a visual representation of the separation process. The peaks on the chromatogram correspond to different compounds, with the area under each peak correlating with the concentration of the compound in the original sample. And then moving forward to step 5, this is when we introduce the separated compounds into the mass spectrometer. So after detection, the separated compounds are directed into the ionization chamber, where they are ionized or converted into charged particles using techniques such as electron impact or chemical ionization. So we have to remember that ionization is essential for the subsequent analysis by mass spectrometry, and it allows the compounds to be manipulated in the mass analyzer, where they can be separated based on their mass to charge ratios. And then for step 6, this is ion detection and analysis in the mass spectrometer. So the mass spectrometer sorts the ionized fragments according to their mass to charge ratio using a mass analyzer. So the two most common mass analyzers would be a quadrupole or a time of flight or TOF mass analyzer. So the detector measures the abundance of these ions. So this step produces a mass spectrum which reveals the mass and abundance of the ions. The unique fragmentation patterns in the mass spectrum help identify the specific compounds that are present in the sample. And then for step 7, the last step, is data analysis and compound identification. So the obtained chromatograms and mass spectra are analyzed using specialized software that compares the data against libraries of known compounds. Pero pwede rin naman siyang i-analyze manually. And then statistical methods may be employed for quantification. So this final step allows for identification and quantification of compounds in the original sample. By matching retention times and mass spectra to reference data, analysis can draw meaningful conclusions about the sample's composition and concentration of its components. So we now move forward to instructions regarding the class activity. So each group will be given a unique data set that includes a premise and specific objectives for analysis. So I need you to follow the instructions to interpret the chromatograms and mass spectra. So for group 1, you will be working on essential oils analysis. So your premise is that we have a local company that produces lavender oils and they want to do quality control of their product. So to ensure product purity, they want you to analyze the essential oil composition using GCMS to check for the presence of key aroma compounds that indicate high quality. So your objectives is to identify each compound in the sample based on retention times and mass spectra, and discuss how GCMS aids in quality control. So you will be given a chromatogram peak, label each compound, and make sure to include in your report how the data would inform the producer about the product quality. So this is a photo of your GCMS results. You will be able to download this in the pre-lab page of this particular module. Okay. Next, for group 2, this time you'll be working with environmental scientists, and they want to check the quality of river water that's close to an industrial zone. So they want to check if there are harmful contaminants that could affect the local ecosystems. So your experimental objective is to identify the compounds that are present in the water sample and then check the relative concentration in each compound and discuss how this can affect the ecosystems in the area and perhaps even human health. So similar to the previous group, you need to label each peak on the chromatogram and interpret the significance of each contaminant's concentration and discuss the role of GCMS in environmental monitoring. So this is the GCMS data that has been assigned to you. Again, you will be able to download this from the pre-lab page of this module. Okay, next for group 3, you will be working on forensics. So this time it's a forensic toxicology case because members of our Philippine National Police suspect that a person has been exposed to certain drugs, exposed with quotation marks. So you currently have a GCMS analysis of their blood sample. So you need to identify what compounds are found in the sample and describe the significance of these findings in the context of forensic analysis, especially in terms of whether they have been taking or have been exposed to certain drugs. So just like the previous groups, you analyze and label each peak in your GCMS data and describe how this data can be used in a court of law to provide evidence of drug exposure. So this is the data set that has been assigned to you, group 3. And once again, you can also download this from the pre-lab page of this module. Finally, for group 4, this time you'll be working with regulatory agencies in the food industry. They need you to test vegetables for pesticide residues in order to confirm consumer safety. So the sample that you have is from a local farm, and you need to check for potential pesticide contamination. So you need to check if the proteins that were found are really pesticides, quantify their concentration, and explain how GCMS assists in making sure that we meet food safety standards. And just like all the other groups, you need to label each peak and the chromatogram, but this time discuss their importance in terms of public health and food safety. So your chromatogram is this, and once again, you can or you will be able to download this from the pre-lab page of our module. So activity schedule here, our meeting one has been diverted into asynchronous because I will be with the Fulbright Philippine Alumni Association today, followed by a meeting with, a presentation at the Congress of the Philippines to present a law that the Philippine Association of Marine Science wants to push. So I'm a little nervous for today. But yeah, that's why I'm not able to attend today's meeting. But for our meeting on Friday, I hope that we can proceed with data consultation. I hope that you can share with me the proteins that you've already identified. And then for meeting 3, which is on Tuesday next week, you can use that time to finalize your report. And if you still need to consult certain parts with me, please message me at least a day before so that I will know to open our Zoom room for consultation. So there is a handout for this activity that's available in Animo Space. Please read it. It can serve as the inspiration for the methods portion of your report, but you can also use methods beyond it. The deadline of the written report is on November 26, 11:59 p.m. For meeting 3, where we will have optional consultation, please email or message me in Animo Space at least a day in advance to schedule a consultation, and make sure that you have concrete questions already and you're not just there to lurk or watch what other groups are doing. And please don't ask me to pre-check your output. Instead, give me concrete options to choose from so that I can give you solid advice. So more or less, that's it for our pre-lab on GCMS. And if you have any questions or points of clarification, please don't hesitate to message me via Animo Space. Thank you so much for watching and see you on Friday.